Okay, so continuing on with DNA replication. Now that we know a little bit about the structure of DNA, let's go into a little bit of background about why DNA would need to be replicated. So when our cells are ready to divide, so remember the main purpose of mitosis, throwback last semester, is growth and repair. So there are many times throughout our lives that our cells probably need to replicate themselves. Anytime you cut your knee, or you scrape your knee, or anytime you're hurt, or anytime your cells die or are damaged, those cells need to be replaced. And so if you're going to do that, if a cell is ready to divide, it has to copy that instruction booklet. It has to copy that DNA and make it, remember how many billion base pairs there are are located within our DNA. It's a lot. So all of that must be copied, and the process of making this identical copy of DNA is what we call DNA replication. So that's why it has to happen. And if you'll remember, this happens during the S phase, the synthesis phase of the cell cycle. So remember, there were 3 phases of interphase. G1, so growing, 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 our cells growing, growing, and then during S phase, our DNA is replicated, and then during G2, it grows, 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 and then mitosis begins. And so, again, DNA replication ensures that each new cell will have exactly the same DNA as that original cell, because we don't want some of our cells having different DNA than the other, because then that's sending a very confusing message. So we want to make sure each new cell has exactly the same DNA. We don't want one cell telling our body to make the protein, so telling our eye to make blue, to make the proteins to make our eyes blue, and then another cell telling our eyes to be brown. You don't want conflicting stuff like that, and so this process is actually pretty foolproof. So the very first step in DNA replication, so if we're actually going to start talking about it, is this. So an enzyme comes into play called helicase, and you can see helicase working its magic here in this GIF. And so we call this unzipping the DNA because it literally looks like a zipper unzipping. And what's happening is, remember, if you will remember those weak hydrogen bonds that I have mentioned over and over and over again in the last video, those weak hydrogen bonds between those nitrogen bases, this enzyme actually has the tools necessary to be able to easily break that bond. So that is step 1. Step 2 of DNA replication, you can't really see it here, but a second enzyme comes into play. Okay, and this enzyme is called DNA polymerase, or the builder, if you want to think about it like that. So DNA polymerase, this should not be the first time that you've heard this term. If you'll recall from a recipe for traits, when we were doing that PCR virtual lab, so remember we were making those billions of copies of that's a very specific targeted sequence of DNA, and we made a bunch of copies. And so remember, they act like tiny machines that read the DNA code and then attach the matching nucleotides to create those copies. And so they bind them according to the base pairing rules. So you can see here these nucleotides kind of falling into play, and they're being matched with their complementary base pair. Okay. So that is the beginning stages of step 2. You can't really see DNA polymerase doing this, but just know those nucleotides don't just magically get there. Okay, and then step 3, the final step of DNA replication that we're going to talk about, is it's finished. So you end up with 2 identical DNA molecules. And we call this process semi-conservative replication because if you think about it, part of the molecule is conserved or saved at the end. So let's kind of take it back a little bit. If we look at the very beginning, so you see how we start off with this original strand. So we have our single double-stranded molecule of DNA, and then at the end of step 1, helicase unzips the DNA, and we have 2 single-stranded molecules. Okay, and so of the old DNA. And then after we add those nucleotides, so what's going to happen? You have this single strand over here. DNA polymerase, the builder, builds an opposing strand to make this strand over here a fully formed double-stranded DNA molecule. However, the strand over here is the original strand, the old strand, and this new strand is one that we just made. So that's what I'm talking about when I say there's an old and a new strand. And the same thing happens on the other side. So remember, we had those two single strands. DNA polymerase adds the nucleotides on this side, adds the nucleotides on this side, and then you have two old strands and two new strands, both linked together. Okay, and so scientists just came up with a word for it because there's a word for everything, and we call this semi-conservative replication.